Hello, this is the Meiosis Lab from Monday, October 29th. So, if you recall the previous week, we covered the topic of mitosis. There's stages of mitosis. You have interphase, which if we split hairs about it, is not technically part of mitosis, but interphase is a stage in between cell divisions. In between. During this time, the DNA is uncoiled as chromatin, long thin threads filling the space of this nucleus. The stages of mitosis are then prophase, metaphase, anaphase, and telophase. Okay, we covered these last week. During prophase, you're going to get formation of the chromosomes, breakdown of the nuclear envelope. During metaphase, chromosomes are lined up individually around the equator of the cell. During anaphase, the centromeres split and the sister chromatids are pulled away from each other, giving that V shape to each of these single-stranded chromosomes. During telophase, the chromosomes reach opposite ends of the cell. They begin to uncoil back towards chromatin, and new nuclei begin to form, and you get the process of cytokinesis. Cytokinesis is division of the cytoplasm. Okay, what we're looking at this week is process of meiosis. Meiosis is the form of cell division that's involved in sexual reproduction and will reduce chromosome number by 1/2. And you notice right here that obviously you have more stages. We go through prophase 1 through telophase 1, then prophase 2 through telophase 2. So a little bit more complexity to process meiosis. Okay, notice that during this process of meiosis, one of the big differences we're going to have is the chromosomes they pair up with their homologs. This is different from mitosis. Mitosis, each chromosome acted individually. Here you have a double-stranded chromosome paired with another double-stranded chromosome that is the same size, same structure, and carries the same genes. And within this pair, one of these chromosomes came from dad, the other came from mom. Okay, as we go through this first stage, the paired chromosomes line up across the equator of the cell. This is metaphase 1. Anaphase 1, the entire double-stranded chromosomes separate from each other and are being pulled away. Then telophase 1, the double-stranded chromosomes reach opposite ends of the cell, and we get cytokinesis. Okay, during this first part of meiosis, these cells are diploid, meaning we have 2 chromosomes in each pair, or we have pairs of homologous chromosomes. So, pair of homologous chromosomes, we've got 2 homologous chromosomes in the same cell. We have 2 homologous chromosomes in the same cell until this completely divides. So, interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, cells are 2N or diploid. Then we go into the second division. We have 2 cells going through this simultaneously. Each of these cells is haploid. It has half as many chromosomes as what we started with, and we have 1 chromosome from each homologous pair. Okay? Second division of meiosis looks a lot like mitosis, but there's half as many chromosomes. So, prophase 2, chromosomes are in the region of the nucleus. Metaphase 2, chromosomes are lined up individually. Anaphase 2, the centromeres split. The sister chromatids are pulled towards opposite ends of the cell. Telophase 2, chromosomes reach opposite ends of the cell, begin to uncoil back towards chromatin, new nuclei form, and cytokinesis occurs. Okay, so on the quiz, you'll need to be able to identify these different stages of meiosis. Go to the next slide. We've got these terms that was just using diploid and haploid. So you need to be familiar with these 2 terms. You need to be thoroughly familiar with these 2 terms. Diploid is also stated as 2N, 2 sets of whatever number of chromosomes constitutes one complete set. In humans, we have 23 chromosomes making one set. In corn, there's 10 chromosomes making one set. In fruit flies, there's 4 chromosomes making one set. So, humans, the diploid number would be 2 sets of 23 or 46. In the fruit fly, diploid would be 2 sets of 4 or 8. So, the diploid number refers to cells with homologous pairs of chromosomes. It's going to be the somatic cells, which soma means body. It's going to be all the cells except the egg cells and sperm cells. The other number that we have is the haploid number. Haploid number is represented as 1n, 1 times whatever number constitutes one set. So in humans, this would be one set of 23. In corn, it'd be one set of 10. So cells with one chromosome from each homologous pair of chromosomes. So the gametes are haploid, and the gametes, that's just a fancy term for the sex cells or the egg and sperm cells. Okay. And N, that is the number of chromosomes that we have in the haploid cell. For example, 23 in a human, 10 in corn, 4 in fruit fly. Now we've got these this mitosis versus meiosis. So let's look at that. Last week we looked at mitosis. This is a form of cell division that is involved in the growth and repair of an organism. It produces the somatic cells, and it's a form of cell division that's also involved with asexual reproduction or cloning. Okay, with mitosis, there are no changes in the chromosome number, no changes in genetic information. Daughter cells are exact copies of the mother cell, and there's only one division. It produces 2 cells. We start with a diploid cell. We end up with a diploid cell. Process of meiosis has one function, and that is it is the form of cell division that's involved in sexual reproduction. It will produce the egg cells and sperm cells, and it's going to reduce the chromosome number by 1/2. In meiosis, the daughter cells are genetically different from each other as well as different from the mother cell. We're going to have 2 divisions. The first division will start with a diploid cell, and we'll end up with haploid cells. It'll reduce the chromosome number by 1/2. The second division will separate the sister chromatids. We're going to produce a total of 4 new cells, starting with diploid cell, ending up with 4 haploid cells. Here's photos showing the process, and as you can see, it's a little more involved than what we had with mitosis, and so we're actually not going to look at the stages of mitosis, although you need to be familiar with that. You need to be familiar with those stages because you will have some questions on the quiz about the stages. Instead, well, let's go quickly through this right here. We've got interphase, where the DNA is interphase, just like interphase prior to mitosis. DNA is uncoiled as chromatin, long thin threads filling the space of this nucleus. Prophase 1, the chromosomes pair together, homologous chromosomes pair together. The nucleus dissolves. Metaphase 1, pairs of homologous chromosomes are lined up around the middle of the cell. Anaphase 1, the homologs separate from each other. Telophase 1, homologs reach opposite ends of the cell, and cytokinesis occurs. All of these stages right here, interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, are diploid. That is, that you have pairs of homologous chromosomes in the same cell. We'll go into telophase 2. What we see is right here that we have 2 cells going through this simultaneously, and you do not have homologous pairs. Notice the homolog, this large chromosome, is over here in a completely different cell. So each of these cells is haploid or 1N. Okay, so prophase 2, chromosomes are in the region of what would be the nucleus. Metaphase 2, chromosomes lined up individually around the equator of the cell. Anaphase 2, the centromere split, sister chromatids are separated from each other. Telophase 2, these single-stranded chromosomes reach opposite ends of the cell, and cytokinesis occurs, which results in 4 haploid cells. These 4 haploid cells will go on to become the gametes, the egg cells or the sperm cells. Okay, so here's up here at the top we have mitosis, and so we start with a 2N cell, 1 division, we produce 2 2N or diploid daughter cells. The only difference is we're going to change from double-stranded chromosomes to single-stranded chromosomes. Process of meiosis down here, we are going to start with a diploid cell. Homologous chromosomes pair together. During the first division, we separate the homologs, reduce chromosome number by 1/2, change from the diploid down to haploid cells. The second division, we're going to separate the sister chromatids and end up with 4 haploid daughter cells. Those 4 haploid daughter cells are the cells that go on to become the egg cells or the sperm cells. Okay, so we'll stop this video at this point right here, and with the next video we'll pick up with this topic of the ovaries and testes. Okay, thank you.